Thank you and welcome to the multi-component solids in pharmaceutical industry themed webinar series actually, because it is an ongoing session of programs every Saturday. So I will and thank you for the by chance I happen to be the opening speaker. So I will cover what this area is about and where it stands today and what are the approaches as well as the opportunities in modulating solubility, permeability, bioavailability, and drug pharmacokinetics using crystal engineering. So just a brief overview. Drug discovery is a very long process. It takes more than 10 years to put a drug in the market. And once the drug molecule is selected by the drug discovery team, which is the chevron arrowhead on the left side, which is in brown color, the drug molecule, which is optimized, is handed over for pharmaceutical development, which is finding the optimal formulation which will make it into a tablet. And the advantage of the crystal engineering approach is that the approaches are so directed towards modifying the pharmacokinetic properties of the solid formulation, that this can be done without changing the molecular structure of the drug. Because as those of us who work in this area, and almost everybody is a subject participant here, will know that one cannot change the even a single atom on the drug molecule once it is discovered and optimized and the approval has been taken. So keeping the molecule intact, how we can improve its delivery profile in the human body is the goal, and that is where pharmaceutical development through co-crystals and crystal engineering comes in. The subject has a great opportunity today, as seen in this histogram bar. You see the classification of the 4 BCS class drugs, which goes to amidon in the Professor Amidon in the early to mid 90s. So BCS class 1 is high solubility, high permeability. Class 4 is low solubility, low permeability. And class 2 and class 3 are the mixed variety, that is low solubility, high permeability is class 2, and class 3 is high solubility, low permeability. And the approaches of crystal engineering, co-crystals, polymorphs, salts, eutectics, solid dispersions, coamorphous solids, these can target the BCS class 2 and class 4 drugs, that is those which have low solubility and either low or high permeability. Now you will see on the left hand histogram that the low class 2 and class 4, which is red plus blue segments of the bar, are about less than 40% in the marketed drugs. But those that are in development pipeline and likely to be launched, that have been launched in the last 4 to 5 years, and we'll see some examples. This number is all more than double that. It's almost 90%. And the word is that the actual number is even more than this. This is about almost 6 years old today. The difficulty of overcoming the solubility challenge is one of the major problems in the pharmaceutical industry, and this is where the role of crystal engineering comes in very nicely. So whatever approaches we take is going to address the solutions to a very large segment of the drug, more than 90%, and that is why this field is growing in importance, as we will see in the forthcoming slides. The area is very bullish today. The largest number of drugs were approved by the US FDA, which is 53, and I think about 6-7 months of this calendar year have passed, and I think the number is more than 35 as of July-August 2021. So the total number at the end of 21 perhaps will exceed 53, which is again a record for the US FDA. Of course, this is a time when we know about the COVID drugs, and this is a very fast moving area for those, particularly the youngsters. This is not like astronomy or space science where you can take a project and try to solve it in the next decade. You have to solve your problems in a matter of months and weeks. Remdesivir and favipiravir, at least 10-15 papers have appeared within 2021, solving the polymorphs, co-crystals, including using electron diffraction for remdesivir and favipiravir. So this is more like T20 cricket. You have to find your drug and you have to find the answer, and of course, very quickly go for publication. Don't sit on the result for too long. And as I am saying this, I think I try to tell my students also the same thing. Don't sit on a result for too long. So the BCS classification, is 1, 2, 3, and 4, and class 2 is the most important category for improving pharmacokinetics, that is low solubility, high permeability. And here in solubility, there are two factors. One is solubility, which is a thermodynamic parameter, and the other is dissolution rate, or the rate at which the peak value is reached, which is a kinetic parameter. And to distinguish between these two, Dressman and co-workers more recently in the last 10-15 years have reclassified 2 as 2A and 2B, where one of them refers to solubility while the other refers to dissolution. And we will see what the differences are, because the approach has to be different whether it is solubility limited as shown in the bottom quadrant, or it is dissolution rate limited. While both are related to how much of the drug dissolves, whether it is the amount or the rate, that changes the approach that one will take to address the solubility problem. These are just some definitions. I won't go into the details, but you can just read it as I'm speaking quickly. Dissolution rate, as I said, is a dynamic phenomena, so it not only depends on the drug formulation, but also the particle size and several other factors. Solubility is defined in terms of the amount of drug that will dissolve in 250 ml of water, because it is assumed that you will take your tablet with a glass of water, which is roughly 250 ml. And that also defines the dose number, which is the number of glasses of water needed to completely dissolve the drug. That is called as D0 dose number. Okay, and then the next is the permeability, or how well the drug binds in the gut or the lumen once it crosses the intestinal phase, and that is another issue in the bioavailability problem solving. That is, once it is delivered, how well it binds to the drug target. The GTT, that is gastrointestinal transit time, these are important because when you are doing dissolution experiments, you have to keep this 6 to 8 hours in mind. So it's about 6 to 8 hours for food, but drugs typically we consider 3, 4, or maximum 5 hours, because after that the metabolic transformations of the drug take over. So these are the different both single and multi-component crystal forms, both crystalline and amorphous. The crystalline are on the left-hand side, and the amorphous ones are those that refer to the solid solutions, eutectics. That is less of crystalline and more of the only the short range order, long range order component is missing, is on the right-hand side. So you can have polymorphs, which is different crystal form arrangements as shown for the polar packing as well as the centrosymmetric packing, or it can be just totally loosely bound a supramolecular arrangement which is not periodic. The multi-component crystals can be solvates, hydrates, salts, co-crystals, solid solutions where the ratio of the two components varies, whereas in solvates, hydrates, co-crystals and salts, the stoichiometry is generally a fixed amount, whether it is 1:1 or 1:2 or 2:1 between the drug and the salt former or co-former. One can also have a slightly discontinuous arrangements, as we see in solid solutions and eutectics. So essentially it is a gradual transition from a completely crystalline state on the left to a completely amorphous or co-amorphous state on the right, with solid solutions and eutectics somewhere in between. This is an important slide, and the message I want to convey is that you can take up the development of pharmaceutical solids, both multi-component and single component, in two ways. One is you want to develop an approach, that is, how can we use co-crystals to solve solubility challenge. So there you would look at those co-formers which can form co-crystals with drugs. And the point I want to convey is that the pharmaceuticals appear in any one of these 6 or 7 categories, which I showed you on the previous slides. They can be polymorphs, solvates, hydrates, salts, eutectics, and the most recent addition in the last 5-7 years, thanks to the advances that happened over the last 2 decades, are co-crystals. The other approach, which is more true for those who are coming from the pharmaceutical industry, is that your drug molecule is fixed. you have a drug for which you want to improve the solubility. Then you can try any one of these approaches and see which one gives you the best solution. So you can either develop a methodology, which would typically happen in academic labs, where your flexibility of which API you select is somewhat wider. On the other hand, in industry, your API is fixed, but you have a menu of options. You can look at any one of these solid forms, and any one would be pharmaceutically acceptable, so long as, and I have not added here several other approaches such as nano formulations, polymer additives, and so on. So you have about 8 to 10 different ways in which you can improve the solubility of these drugs. So our group, this was already mentioned, so I will not spend too much time, looks at various APIs in terms of how to improve their solubility. We also want to study the state of these APIs when they interact with a coformer, and that's very important from a not only from a solid form structure perspective, but also from a regulatory side. It's very important to know whether the acid-base complex is a neutral co-crystal or an ionic salt, or it's actually a quasi-continuum state or a quasi-proton state, and the salt co-crystal continuum is present. And this there are many methods known, and we have added the XPS techniques. So we look at co-crystals, we look at polymorphs, we look at co-amorphous solids, and we also look at drug-drug combinations on how the crystal engineering approach can be used to improve the bioavailability, stability, and the quality of the pharmaceutical formulation. I will not go into the details of how we make these, but just so that those of us who are a little outside the area will know how these are done. I will not repeat it for every example. So we make them either by solution crystallization, which is not the most preferred method, but is always used. We can do it by grinding, which can be either manual or mechanical, sonication, spray drying, crash cooling, lyophilization. There are many methods used. Ultrasound, hot melt extrusion, rotovap solvent evaporation at a fast rate. Essentially, when you change the conditions, you change the energy landscape of crystallization and therefore the nature of the product that we. Once you get the product, we analyze it by diffraction technique, microscopy, spectroscopy, Raman, infrared, NMR, and then we also look at the thermal analysis, the surface characterization, as well as the particle size. As I mentioned earlier, particle size can be very important for the dynamic phenomena such as dissolution, as well as the way in which the pharmaceutical will bind to the solvent water molecules and so on. So here comes the rational design part of crystal engineering, and here I have shown different synthons, and heterosynthon and homosynthon is the nomenclature used. Homosynthon is the hydrogen bond dimer motif between two functional groups which are the same, which is acid-acid or amide-amide. So that's referred to as a homosynthon. Heterosynthon is between unlike functional groups, which is carboxylic acid, pyridine, or carboxamide and acid, and so on. The percentages below are just a rough guide of how excellent or how good or how moderate is the probability of their occurrence if these two functional groups are present in the molecules. And that's important because it gives you a quantitative confidence of getting this target synthon if you took a carboxylic acid and a pyridine, or a carboxylic acid and an aminopyridine, which are very common functional groups in pharmaceuticals. On the other hand, if you have an acid amide, then you only have a 1 in 2 chance of success rate. Compared to that, acid-acid and amide-amide are just 1 in 3. So it's all relative because competition is happening between all the functional groups when you carry out a dynamic crystallization. And as we all know, the supramolecular synthon idea was advocated and proposed by Professor Gautam Desiraju in 1995. And since then, crystal engineering, which was largely a structural chemistry subject, actually entered the domain of organic chemistry and subsequently inorganic and materials chemistry. And its maximum impact has been in the pharmaceutical sciences or pharmaceutical chemistry area. The next, yeah, sorry. I think somebody's microphone is on. Okay, yeah. Am I audible? Am I audible?
Yes, sir.
Yes, sir.
Okay, thanks. Yeah, so the next major step in this pharmaceutical co-crystals happened roughly less than a decade after the synthon idea of Professor Desiraju. That is, Professor Mike Zavarotko wrote a very seminal review in ChemCom in 2003, where he showed that these synthons can actually be the building units for improved pharmaceuticals, and that was the time when this heterosynthon idea, that is a synthon between two unlike functional groups, which is required for a co-crystal, because a co-crystal will have two molecular entities. So you need two different functional groups, say a carboxylic acid and a aminopyridine, or a carboxylic acid and an amide, and that essentially set the stage for what has happened in the last 15 years, and that's what we are going to take a quick overview of as we move along. So this is essentially a sharing of the timelines, starting 1995 with Desiraju's review, then 2003-4, as I mentioned, the ChemCom review. Then pharmaceutical co-crystals became a mainstream of pharmaceutics, improving the tabletability, and I would say in 15 years from the start point roughly, which is taken to be the beginning of the millennium, 2000, we had the launch of the first salt co-crystal drug, Entresto, which is a relatively fast timeline in going from what we call as lab to market. And subsequent to Entresto, there has been a large number of pharmaceuticals which have appeared in the market or are in development pipeline, which have been developed using a rational crystal engineering approach, and not I would kind of draw a line between the co-crystal salts which are developed using crystal engineering approaches and the mere salts which were already there in the pharmaceutical industry. The most preferred form of a drug formulation is the salt. They were already there, but now we have, as I just showed, a hydrogen bond directed approach to making pharmaceutical formulations through co-crystals and eutectics and co-crystal salts. That one can say the timeline is between 2000 or 1995 2000 to about 2015-16, and it continues. I think every year at least one or two salt co-crystal drugs are coming out of the development pipeline. So these are the poster examples of salt co-crystal drugs, and they could not have hit the market as a formulation unless they were formulated or developed. The pharmaceutical development happened as a drug salt co-crystal for Entresto, and as a drug co-former complex or a co-crystal for etugliflozin. So basically, the crystal engineering result or the crystal engineering approach was essential to the formulation. They could not have kind of been made into medicines from a molecule which is a drug. They could not have been made into medicine tablets and powders unless the methods and approaches that I have just described were used to discover the best optimal formulation. So this is just to show for those because this is an interface between, as we just heard in the introductory remarks also, between drug development and the in vivo study. This is the first study on LCZ696, which is a code name for Entresto, and this it's a cardiovascular drug. This shows the advantage of using the drug-drug combination, that is enalapril, which is valsartan, and sacubitril, which is another drug molecule. The drug-drug combination of enalapril and sacubitril, which is Entresto, is about 20% superior, because this refers to the mortality rate. So the mortality rate between those who took the single drug is about 20% higher than those who took the combination drug, and that's quite a big percentage, considering that now we are seeing the numbers and the percentages related to drugs and pharmaceuticals almost on a daily basis in newspapers. So an improvement of 10%, which is mortality rate 914 versus 1117, and it's a very long study lasting several years on a cohort population of more than 4,000 patients. So it's very robust data, which clearly shows the advantages of the salt co-crystal drug-drug combination compared to the single drug alone, which is already on the market, that is enalapril. Several others, as I said, have been following these trailblazing early leads. These are just some of the examples, and we are adding more as we speak. As I said already, more than 30 drugs have been approved by the US FDA in this year itself. So I will now share a few examples of what approach one can take to improve the solubility and permeability. And these are some of the, I will not say that these are rules, but these are more like guidances. These are, directives which one can use to be on the right track. So it's like if you are going hiking, then those who have gone before you, they usually set a trail. And if you are an early hiker, you just use the trail. You don't, just wander into the mountains or the jungle. So this is like a trail which has been laid by those who have worked in this area over the last 20 years. You are free to use your approach, but this is a trail which seems to work. So one can use this and then, of course, build on it, as I say, similar to what you would do in a mountain or a jungle hike or a picnic. So hydrogen bond synthon probability, I have already shown you that you can use those numbers, which can be derived from the Cambridge database, which is now over 1 million crystal structures. So you get a large amount of data for the drug of your choice because you have a diverse set of structures in the database. The delta pK guide is a very important one because it tells you whether you will expect a salt or a co-crystal, and this guidance, delta pK guide, is also used. The term is also used in the FDA rule book on pharmaceutical co-crystals. So 0 to 3 was the original band for the mixed state, that is salt co-crystal state, and less than 0 delta pK means a neutral co-crystal. Greater than 3 delta pK means it's quite large difference between the acidities and basicity, so it will be a salt. But now, based on the data we have from a large number of crystal structures, almost 10,000 crystal structures of acid-base complexes, this has been actually expanded to -1 to 4. So -1, 0/0 is the neutral side, and 3 to 4 is the ionic boundary line, and anything in between this is like a mixed call. It's a gray zone. One can also use computational models, and there are several softwares available for screening those solvents which are most appropriate for the drug and the co-former. The next is a very important one. It's an empirical parameter, but very useful, the Hansen solubility parameter, because that allows you to estimate the extent of mixing that will happen between the API and the co-former. And the mixing is important because mixing allows you to induce or promote reactivity. And this can be derived from, it's a very classical parameter, almost more than 50 years old, but there are several improvements to it. And this can be, it's essentially a combination of hydrogen bonding, hydrophobic, van der Waals, ionic interactions, but there are certain weightages given, and that allows you to derive these solubility parameters from the structure of the API or the drug or the co-form. The last is, of course, very important because when you work in this area for a while, then you get a certain feel for it as to what would likely be working for the kind of drug you are dealing with. And that's true for any research area, also true for this area, that if you already have familiarity with a certain class of drugs, whether it is diabetic or anti-cancer or cardiovascular, you will have a certain preference of what you think is likely to work. So you can use a combination of these. As I said, it's not limiting, but it's more of illustrative. So we will use these ideas when we look at some of the examples here. The first example is etrodolac, which is a NSA drug, which we took as a model compound. And the reason why we took it as a model compound is A, it has low solubility. B, the dosage is very high. So therefore, the dosage is high because the solubility is low. And so if we can improve the solubility, as you can see, it's a huge tablet. And, twice daily, if a patient has to take such a huge tablet, compliance is always an issue. And those who are unwell find it very difficult to swallow large tablets. And so therefore, improving the pharmacokinetics or solubility permeability of such drugs is always more important than taking those for which the dosage is low because the tablet size would be generally be much smaller. So again, we come back to PK. As I just said, this is this is a racemic drug. It has a chiral center, but the drug is a racemate and it has a carboxylic acid group. So therefore, the choice would be to look at bases with a certain pK, and the carboxylic acid has a typical pK of 4 to 5, which is normal for a carboxylic acid. And we looked at several amines, and the idea was to scan basically aliphatic, both cyclic and aliphatic amine, and see what kind of A result we get, whether it's a salt or a co-crystal, and B, the improvement in solubility and dissolution. And the third important thing one you want to see is whether the salt or the co-crystal complex, whether it is stable to the conditions of the solubility and dissolution experiment, or it sort of dissociates and decomposes. Because if it dissociates or decomposes, then you have to look for something else, even though it may have very good pharmacokinetic properties, because you need an optimization of both improved solubility and good stability. So you can see that the delta pK value is quite high. It is more than 4 in all the cases, as expected with amines, and all the products are indeed salts. So this is to show it's a crowded slide, but I just want to convey a quick message. So I will just highlight what I want to show. This is on the left top is the crystal structure of the API, where you see the NH and the CH group in a bifurcated hydrogen bond synthon bonding with the carbonyl group of the carboxylic acid, and the OH of the carboxylic acid is in a trans conformation and intramolecularly hydrogen bonded. So essentially, when you make a co-crystal or make a salt, one is going to break these strong hydrogen bonds which are there between the drug molecules, and interrupt with either an ionic or neutral hydrogen bonds which are stronger than those which were there. So there is also an enthalpic factor when you are going from the single molecule crystal structure to the co-crystal or the salt structure. And here the counter ion is an ammonium species, or a alkyl ammonium species to be precise. So this is the isopropylamine. I'm not putting the R group of the amine just to make the structure clearer. You see that the carboxylate anion sits in the middle, and it's hydrogen bonded to the large number of oxygens which are present in the drug molecule. So essentially, the drug molecules surround the ammonium ion in the isopropylamine, and similarly the hexylamine, N-hexylamine, and cyclohexylamine, phenethylamine, and piperazine. So all these are in one way or another NH groups which are hydrogen bonded to the drug. They have, each structure is slightly different, but the essential point is that it has strong ionic protonated nitrogen NHO hydrogen bonds, O or O minus hydrogen bonds. Now comes the property profile, which is what we would like to enhance, and that is the main goal of the structural part. Is it sits as a information for the crystal engineer, because that tells us as to what the structure is, because then when we see the property or improvement or not an improvement, as we see in this case, then one can try to correlate the property and the structure, because that gives us a model to explain what is really happening within a system. So here we find something which is quite unusual, that all the salts are little higher than the parent drug, which is at the bottom. You see these straight lines, except one, which is the isopropylamine, which is showing almost a 20 times enhancement compared to the reference API. The reference API is right at the bottom, and all the other salts are somewhere little higher, anywhere between 5 to 8 to 10 times, and more than 20 times improvement is the isopropylamine salt. And these in resolution rate studies were carried out in phosphate buffer. So this is carried out, as you see, over 3 hours. As I said, this time interval is typically between 3 to 4 to maximum 5 hours. One doesn't need to go beyond 3 to 4 hours. And this is the cumulative drug, which is amount of drug that is dissolved, because over a period of time it will build up. That again is the highest for isopropylamine. And this is how fast or how slow is the peak concentration, or the Cmax, and that is about 40 minutes to 60 minutes. This can also be less, and you want to know this because that tells you when the peak drug will be delivered, because that's important for a patient, because that's when the maximum therapeutic effect will happen and how quickly it will happen. And for an NSAID, you would want it to be relatively fast because you want instant relief. So essentially our results which have been published in Crystal Growth and Design, and we can any more details we can discuss offline. So both the intrinsic dissolution rate and the diffusion rate, which is across a semi-permeable membrane, they increase for the same salt, and that's nice because when both the solubility and the flux rate or the permeability are increasing for the same salt, that means then one can really select that as the best salt. Now, why this happens is because there are tight ion pairs, acid-base ion pairs, which are present between the carboxylic acid API and the ammonium salt. And the reason why we believe so the solubility increases because of the ammonium salt, and the low lipophilicity of the isopropylamine compared to the long chain and the cyclic amines is responsible for the high permeability as well, because this is already a relatively large molecule. So if you put a very large coformer or a salt former, the transport across the semi-permeable membrane will become slow because of the very bulky size of the molecule, of the drug molecule, and the salt former or the coformer hydrophobic groups. So the balance or the ideal R group is a small alkyl R group on the amine, and the ammonium salt increases the. So essentially we have both solubility and permeability increase for the isopropylamine salt, whereas the rest of the salts are only of moderate improvement. The next is an anti-cancer drug, dabrafenib, which was more recently approved for melanoma cancer, and it is also used as a combination drug. This is marketed as a mesylate salt, and it is a BCS class 2 drug. So although it's a mesylate salt, it is still not having optimal or ideal solubility. It is still in the low solubility category of BCS class 2. The characteristic of this drug is that it is zwitterionic. Many drugs are zwitterionic, just like amino acids. So they have a acidic group and a basic group within the same molecule, which means either they can form salts with acidic coformers or basic coformers, or under ideal suitable pH conditions they can also be existing in the zwitterionic state as a function of pH, as happens for amino acids. So essentially this is just to show you that the pKs in the drug molecule are because of the different functional groups vary over a wide range from 6 to about 2 in the middle, and negative pK for the third functional group. So this is the structure of the drug molecule, and as you see, it has a basic part, the aminopyridine part on the right hand side bottom. It has an acidic part on the left hand side up, which is the sulfonamide group. Normally sulfonamide group is not acidic enough to be deprotonated, but because it is surrounded on both sides by a fluorophenyl group, it makes it electron withdrawing enough that it can be deprotonated. So it can make an anion here of the sulfonamide. It can hydrogen bond to acidic groups on the aminopyridine side. So this is indeed a zwitterionic molecule, and the mesylate salt means that the salt is forming with the aminopyridine group, which is the basic side of the molecule. And so we have in this oval shaped diagram, we have subdivided the coformers as acidic coformers, which are on the top side, which is methane sulfonic acid, fumaric acids, and various other carboxylic acids, which would hydrogen bond typically to the basic functional group on the molecule, that is the aminopyridine. On the bottom are the amines, which have not been explored so far. The acids have been explored. The bases can hydrogen bond and deprotonate the sulfonamide part of the molecule, and therefore make again either a co-crystal or a salt depending on the delta pK difference. So the bottom coformers are what we have explored in this study, and we are going to compare with the mesylate salt, which is already the accepted formulation for dabrafenib in the tablet. So this is where the solubility dissolution factor comes in. you will see that the third column is essentially unfilled here, which is equilibrium solubility. That means equilibrium solubility is typically measured at about 12 hours, beyond 12 hours, typically 12 to 18 to 24 hours. And therefore, this is unfilled because all the salts are not stable for a time long enough in the slurry medium of the experiment. So we only look at dissolution rate or IDR, which is the parameter that we measure in a shorter time frame of 2, 3 or maximum 4 hours. So and this essentially the first row is dabrafenib, which shows the IDR rate as 27 milligram per centimeter cube per minute. So it's a dynamic rate. The mesylate is about double that value, 42. We kind of just leave the rest of the rows. We go to the last row, which is 227 for the ethylene diamine hydrate, which essentially is about 10 times that of the API and 5 times that of the mesylate salt. The others are either lower or higher relative to the API, and we kind of don't worry about them for the time being. Sorry. Tarvata. So this is the dissolution rate, which shows the ethylene diamine salt very similar to the previous case of etodolac, where the maximum solubility and dissolution was for the isopropylamine salt. Here it is the ethylene diamine hydrate, which is showing the maximum profile, and all the other cases are relatively close to the baseline of the API. And again, now we will see with the crystal structures why the ethylene diamine hydrate is showing such a dramatic improvement in dissolution rate. So these are the these are the crystal structures. Sorry, let me go back to the pointer view. So this is the crystal structure of the mesylate salt on the left top, where you see that the methane sulfonic acid hydrogen bonds to the aminopyridine, as I had mentioned in the introductory part of this system. This is the isopropylamine salt, and this is the ethylene diamine salt. We straight away go to the ethylene diamine salt, which is a hydrate and which has a hydrogen bonded chain between the between the water molecules and the protonated ethylene diamine. And when you look at the crystal structure of the salt part, that is, you see that that the nitrogen of the sulfonamide group, the proton is not present here. It is actually present on the ammonium part. So you have an ethylene diamine salt, deprotonated sulfonamide group, and this forms a cyclic hydrogen bond synthon between the protonated amine of ethylene diamine and the deprotonated sulfonamide group with NHN minus hydrogen bond and NHO hydrogen bond with the sulfonamide group. So this is a cyclic synthon, and this is a chain synthon or a chain hydrogen of hydrogen bonds between water and ethylene diamine. And so as the essential difference between the mesylate salt, which is our reference comparison, which is the hydrogen bonding between the aminopyridine and the sulfonate group, and on this side the ethylene diamine and the sulfonamide group, we try to now explain why the ethylene diamine salt is superior to that of the mesylate salt. And our explanation is that there are two explanations. One is that the hydrogen bond chain, which includes the water molecule, essentially will drag more water molecules into hydrogen bonding with the ethylene diamine because of the electronegative NH2 groups present. The second is the aminopyridine group, which was hydrogen bonded for the mesylate salt, is now relatively free. It is not hydrogen bonded in the ethylene diamine salt. So this NH2 and pyridine groups of the API can very nicely and strongly hydrogen bond with water molecules. So there are two sites on which water can hydrogen bond with the API in the ethylene diamine salt. One is the aminopyridine side of the API, and the other is the hydrogen bonded chain between the water and the ethylene diamine. And because of this large number of hydrogen bonds between solvent water and the salt, the solubility of this particular ethylene diamine salt is far superior to that of the known mesylate salt. Are we doing well with the audio and the visual?
Yes, sir.
Yes, sir.
Fine. I like to check because sometimes it just goes off or goes offline. Okay, looking at the time, it's 9:36, so I will try to wrap up now. The next example is essentially a continuation of the previous one, so I will not go into the details, but I will just show an overview of what the idea was. Now we look at some zwitterionic drugs, and the idea of looking at zwitterion drugs, which is the fluoxetins, is that the solubility, permeability are essentially, competing properties, or it's like a seesaw of balance in the pharmacokinetics of drugs. The solubility comes from the hydrogen bonding functional groups in the drug molecule, and the permeability comes is because of the hydrophobic or the lipophilic groups in the drug molecule. And any drug molecule will have alkyl groups, phenyl groups, and also hydrogen bonding groups, as we have seen in the previous two examples, and is well known for pharmaceuticals. So essentially there is a balance between the hydrophobic and the lipophilic portions of the molecule, and that defines the solubility permeability balance of that particular drug, and essentially whether it belongs to class 1, 2, 3 or 4. Our idea was, can we use crystal engineering to tip this balance between the zwitterionic form of fluoxetine drugs to the neutral form, because the neutral form will be expected to be more permeable than the ionic form, because the ionic form will be more soluble but less permeable, while the neutral form will be could also be equally soluble or more soluble because of the coformer, but certainly more permeable. So essentially to improve both solubility and permeability. So the idea here was, just like amino acids, the fluoxetine drugs are also zwitterionic. They have this piperazine ring, which is which is protonated, and the carboxylic acid group, which is an anion, and there's an intramolecular proton transfer, and that's what most of these fluoxetine drugs look like, which is shown on the bottom. The idea was, if we hydrogen bond these fluoxetine drugs with a coformer, the OHN hydrogen bond, which we know is a very strong synthon between the hydroxyl donor and the nitrogen acceptor, we felt could perhaps replace this protonated nitrogen and make it a neutral nitrogen, and as a result this proton, which is on the piperazine ring, will end up on the carboxylic acid, making it into the neutral form of the drug rather than the zwitterionic form. And so we looked at the hydroxy benzoic acid ester, or the paraben esters, the methyl, ethyl, and isopropyl and propyl, and so on, and butyl. So this is just to quickly show the result. What we expected did happen. That is you can see that the hydrogen bond OH goes to the nitrogen of the piperazine. That means that NH group, which was protonated in the parent API, is now a neutral group because of this strong OH and hydrogen bond, and the proton, which was on the protonated piperazine, has now ended up on the carboxylic acid to give this intramolecular hydrogen bond. So from a zwitterionic API, here we have a neutral API which is bonded as a co-crystal in a few of the paraben ester derivatives, as I just showed you on the previous slide. Now this is what is really what we want to see. Here we see that the propyl and the ethyl esters of the hydroxy benzoic acid are showing much higher, this is the dissolution rate profile than the reference drug. And we quickly see the solubility, which is good for ethyl and propyl. The cumulative drug, which is the permeability, is also reasonably good for ethyl and propyl. So ethyl propyl esters are showing, just like the previous case, both high solubility, high dissolution, high flux rate, and high amount of cumulative drug dissolved and also crossing the semi-permeable membrane. So the right combination of coformer, and that too in the same coformer when both solubility and permeability improve, that is more desirable, because then you can select that as your next preferred salt or co-crystal combination. This is the spring and parachute model, which I will just briefly refer to, which we have postulated more than a decade ago as to why co-crystals show a dramatic improvement in solubility and dissolution relative to the parent API. Typically, in a parent API, what you see is we refer to as a spring effect, which is shown as the blue curve. That is the, of course, the reference is the drug at the bottom, which shows a very, very low solubility. That is the stable crystalline form. If you have a polymorph or an amorphous state, which will be graph 2, it will show an immediate spike. But as soon as there is supersaturation, immediately precipitation happens, and within about half an hour or 40 minutes, the drug solubility and dissolution crashes down to the parent value or the original crystal form value. So you don't get that much of a area under the curve because of rapid precipitation. What you are doing in a co-crystal is that you are delaying the decline or the drop of this peak solubility, and that is why it's referred to as a parachute. Rather than drop in 20 seconds, you are going to drop over 200 minutes, which is typically what we would like. So that's your parachute, and your co-former is the basis for the parachute effect. So you are extending the metastable zone width from less than 1 hour to about 4 hours, and therefore, the AUC or the area under the curve for drug is dramatically higher for the co-crystal salt pharmaceutical compared to the parent drug. I'll now come to the ending, the last few examples. This is we have used X-ray photoelectron spectroscopy to show that one can study the movement of the hydrogen between an acidic and a basic functional group. And as I said, this is not only important from a structural point of view, but also in terms of defining, because anytime you take a drug formulation forward, one has to clearly specify whether this is a neutral co-crystal or an ionic salt. And that's very important in terms of classifying. And not all drug salts and drug co-crystals are amenable to single crystal X-ray diffraction. So we need orthogonal methods, and the word orthogonal is used in the FDA guidance also. And so XPS is one of the orthogonal techniques. Other techniques are, of course, IR spectroscopy, NMR spectroscopy, and several other methods are there. X-ray diffraction is there, but as I said, X-ray diffraction is not possible every time for pharmaceutical solids. So one would need non-diffraction techniques, and so we have used X-ray photoelectron spectroscopy to study the salt co-crystal movement or continuum state, particularly using XPS. So the idea is, if you have an acidic and a basic group, it can be a neutral co-crystal as on the left, it can be an ionic salt as in the middle, or a quasi-state as shown on the right-hand side. So these are just some model examples of acidic and basic functional groups, and you see that they will give a delta pK in the range of about 0 to 3 or -1 to 4, and that was the starting point of our study. It's not a very large study, but it's an ongoing study, and so hopefully we will get more data, and with more data we will be able to refine this XPS method more precisely in terms of what range is likely to give a neutral or a salt or a continuum state. I'll straight go to the results. Basically, XPS measures the amount of energy required to eject the N1s electron, because the nitrogen is neutral. It will be electronegative at a zero charge state. If it is a protonated nitrogen, it is the same nitrogen which is positively charged, and therefore, because it's got a larger positive charge in the nucleus, the amount of energy required to eject the 1s electron will be higher, and that higher is typically about 2 electron volts. So neutral nitrogens are about 398, 399 EV, whereas sometimes going up to 400, whereas ionic nitrogens, to eject the same 1s electron by XPS, is slightly higher energy, 400.5 to 401, sometimes going up to 402. So it's a very narrow range, but yet you can clearly define the boundary line between what's a neutral and what's an ionic state. At least we hope to. These are the XPS plots. I will, but essentially, because we know the crystal structure also, we know this is more of a model study to parameterize the system. So we know whether the result is a salt or a co-crystal or a continuum, and then we try to calibrate our XPS plots. Those details are there in the paper, so I'll not go into the plots themselves. I'll go straight to the results, which shows that so this is the x-axis is the energy scale, and as you see, the salts have high EV values beyond 400. Typically 400 to 401 is the salt range, as I already mentioned from the table. 398, 399 is the neutral co-crystal. We would have liked to see the salt co-crystal continuum somewhere between these two values. But as I said, this is a small data set, perhaps not the best data set, but whatever we thought was our first set of molecules. We do see the salt co-crystal continuum, but the values are either closer to that of the neutral value or they almost reach the ionic value. If we could get a range somewhere in the middle, that will give us, you know, the defining band for the continuum state. At the moment, we don't have the data, but hopefully with more examples, which are ongoing experiments, we'll get that data with structures which will allow us to find the band between the violet bands and the blue. So the green is at right now. Actually, if I didn't tell you on the right hand side whether it's a salt or a co-crystal or a continuum, if I remove this thing, this information, you will not be able to tell without the color coding whether your product is a salt or a co-crystal or a continuum. And therefore it doesn't have a predictive value. It only has information in terms of data, but not prediction. So to make a prediction, we need an intermediate band, and that's what we are trying to work at. So I'm not saying this is the final conclusion. At the moment, we believe that it's difficult to predict, but we are hoping that with more data, we will get a predictive value for the XPS. Another important area, and I think with the COVID-19, this has become very important, that is repurposing of drugs. So drug co-crystals, because you have multi-target therapies, and through a linker, one can very easily do drug repurposing as well as going into newer therapeutic areas. This is just to flash a few slides. It's not really to go into details. This is an example from one of the companies, which I'm sure those who are from the pharmaceutical industry will be aware, where this anti-cancer drug, the best combination was developed by Teva using in the fumarate salt, which is to improve the stability of the ibrutinib drug. This is another result from our lab, which has appeared in the last 1 week, where we have shown that the carboxylate aminopyridinium group salt, which the aminopyridinium group appeared also in dabrafenib. So it's a very common functional group in drug molecules, makes a very strong synthon with hydroxybenzoic acids, and these are all GRAS coformers. I should have mentioned in the beginning itself, which I overlooked to mention, that the coformers we used in these studies are acceptable molecules pharmaceutically. That is, they are GRAS molecules generally regarded as safe. So hydroxybenzoic acids, which is the coformers in this study, are pharmaceutically acceptable, and it makes a very strong synthon. So the hydrochloride salt of the multikinase inhibitor drug pazopanib is already been studied in terms of its dissolution profile at different pHs. Now we are studying the different hydroxybenzoate salts as well. The last example I want to show is, as I said in the beginning, if your molecule is decided and you want to improve its solubility, you can take any of those 7 or 8 drug forms, pharmaceutical or crystal forms that were represented. So we found that the coamorphous solids with amino acids are excellent ways to improve the solubility of curcumin, which is a bioactive molecule. And here we see that the tyramine salt as well as the arginine hydrate show excellent improvement in dissolution and solubility. So whether it is a co-crystal, whether it is a coamorphous, the idea is to target the property and then study the solid form using the variety of techniques that are already known. I think I will stop here. It's 9:50. I had said 9:45. So essentially, I have tried to show you that pharmaceutical co-crystals and salts provide a path to improve medicines. And I'm just stealing the words from Zavarotko's review about 15 years ago in 2003-4. I think that dream which he postulated was based on the synthon idea of Desi Raju, which came out in mid 90s, I think has been actually realized for the pharmaceutical industry after 2 decades. So we have a very robust chain of pharmaceuticals coming out of the pharmaceutical crystal engineering principles, which we are now familiar with. So solubility, dissolution, bioavailability, we have, I think, good models. I will not say they are predictable, but at least we have a rational approach how to target them given API. Heterosynthons and hydrogen bonding are extremely important. So therefore, we need crystal structures, at least for those combinations or those co-crystal salt combinations where the single crystals are available. And now, of course, electron diffraction allows us to get single crystals of very, very large number of solids which were not possible in the past. And this also has come out in several journals, like compared to paracetamol, orthocetamol does not give single crystals. So that has been determined by X-ray diffraction. So pharmaceuticals which were not accessible in single crystal structure state earlier are now very easily accessible thanks to multiple techniques, including electron diffraction, which is now accessible. Permeability is really is at the same stage that solubility was about a decade ago, I'll say. So while permeability is lagging behind, but I think it will quickly catch up with solubility, which means whether it is solubility or permeability, we can modulate and improve it in a systematic way for drug pharmaceuticals. Last but not the least, I'd like to thank my group of students, postdocs, as well as my collaborators, the group at NCL, Hyderabad University, and the various funding agencies for their generous funding. Thank you very much all for your kind attention and being present at an early hour on a Saturday morning, at least for the Indian participants.